Very good morning to my dear students. Welcome to MVM Science and Home Science DigiLearn. Today we are going to discuss regarding the practicals of paper number 301 of semester 3 SYBSc Microbiology. I, Juhi Shet, welcome you again. In this paper, we are going to study about the microbial diversity. And here, practicals are there. So, which practicals are there? Their list is already with you. From that list, today we are going to discuss about the practical number 8, that is microscopic observation of different protozoa from the given sample. Now, to begin with, what are protozoa? Protozoa are microscopic unicellular organisms that have complex internal structures, right? They are mostly not photosynthetic by nature, so they are not autotrophic. They may be heterotrophic or they may be holozoic by the nutrition. Here is the structure and classification of the protozoa. Classification is done into two different phyla, that is Sacromastigophora and Epicomplexia. Next, life cycle stages. Now, very important thing about protozoa is that during their whole life cycle, once they need to do the process of cyst formation. Cyst, we all are knowing that are the structures which are found external to cell wall for protection. Reproduction, they mostly do sexually and asexually, but generally they do or they reproduce by binary fission. Nutrition is, as we discussed, maybe holozoic. These are the various pictures of the protozoa when observed from the microscope. As our practical says, that microscopic observation. So in microscope, how different protozoa looks, here is the picture. This practical has certain things like you can do it or you can study or observe with the help of the microscopic observations, that you can do the use of permanent slides also for the study of protozoa, or you also can prepare the slide from the given sample and then you can mount it in the microscope and observe. But before proceeding for that, let us understand few of the characteristics of protozoa which are very much common and useful. They are unicellular and some may be colonial. Colonials means they may be staying in groups. Most of them are microscopic, all are symmetric, and they are within the members of the group. No germ layers are present, no organs or tissues are formed. They include free living, mutualistic, commensal, and parasitic forms. They move or they do the movement from one place to another with the help of various organs like flagella, pseudopodia, cilia, etc. They may have endoskeleton or exoskeleton, but it is very primitive, or you can say in a simpler form. They may be aquatic or terrestrial. And reproduction is done generally by the binary fission or budding or sometimes cyst formation, or if it is in the form of sexual reproduction, then either by the conjugation or syngamy of the gametes. So these were few of the characteristics of the phylum Protozoa. Now let us understand how we will be preparing slides. But before that, you should be aware of the few names of the protozoa. So here are some of the medically important protozoa which are responsible for either causing disease or in some or the another manner. So they may be medically important. Now, here is the skeleton of the microscope. You all are very much familiar with the microscope, how it works, just because in this practical we have to do microscopic observation. So to be clear that how we will place. So here on the stage, we are going to place either the permanent slide or the prepared slide, and then with the help of the focusing. Here focusing will be there, then we will be able to observe through the eyepiece the protozoa what we are going to study. Now let us understand a few of them. There are large varieties of the protozoa, but here we are going to discuss or understand only few of them. But just to make you aware, here is the figure of various types of protozoa. Now, for performing this practical, what all things you require? You require first of all the sample which contains protozoa. So from where you can get the sample? So sample can be from various types of rivers, lakes, ponds, rocks, or stored water. Sometimes in our home or our building also stored water or storage tanks of water are there. From that also you can collect the sample. Then you require a clean, non-greasy glass slides, cover slips. We require microscope, or if we want to observe the slides from the permanent slides, then we require the permanent slides. So these are the general requirements. Then what is the procedure? How we have to make the slide? First, we have to take a clean non-greasy glass slide. Then we have to mark, as we know how we can mark in the slide, we can label in the slide. And then we have to put a drop of the sample or water, right? And then we have to place the cover slip and observe it under the microscope. Now, how you have to observe the permanent slides? Permanent slides can be observed with the help of the microscope, of course. You have to place the slide, permanent slide, in the stage of the microscope, and then you have to do the fine and coarse adjustments, and then you have to focus, and then you have to observe. After observation, we need to draw and conclude our things, that what all things we have observed. Let us discuss first protozoa, that is amoeba. Now we all are very much familiar with this word amoeba. Here is the structure of the amoeba, amoeba proteus, right? Amoeba is having a very irregular structure with the finger-like projections. We usually call it as pseudopods. Then there are nucleus, and here is the cytoplasm. This is the very basic structure of the amoeba. Here you can see the process of binary fission, which you have already studied in your previous standards also, that understand and study the diagram of the binary fission, how binary fission takes place. So a parent cell is there, then it is forming a pseudopod, then the nucleus is being enlarged, then after that the nucleus and the cell will be pushing or moving away from each other, and then finally they both will be separated, and then two daughter cells will be formed. So from one single parent cell, two daughter cells are formed. Next, amoeba do not have cell wall. They are generally stained pink in color. Then they are feeding by the help of the pseudopodia. So sometimes here, if food material is there, or your food material is there, they will form the pseudopodia, and they will engulf the food into it. Here you can see a clear diagram. This you can even draw in your book, so that you will get a clear idea, or in your journals whenever you are being provided with. So this is the structure of the protozoa. Here you can see a food vacuole is there for the engulfment of the food and digestion of the food. Here is again one more picture of the amoeba, how it looks from the electron microscope. This is the process of again phagocytosis. Next protozoa which we are going to study is Plasmodium. Plasmodium name uh, uh, call, says us that it is one of the reason for causing the malaria, because we have studied in our previous years that malaria is caused by the protozoa Plasmodium vivax and Plasmodium falciparum. So Plasmodium is the genus of unicellular eukaryotes that are obligate parasites and vertebrates uh, and insects. The life cycle of the Plasmodium species involves the development in a blood-feeding insect host. Either it may be animal, human, or other insects, right? They grow within the body tissue, and they do the destruction of the host blood cells, especially the RBCs, causing the disease malaria. So this is in general about the Plasmodium. Plasmodium are found all over the globe, all across the universe, right? It is very important to understand that for their completion of the life cycle, it is important that they do a attack on some host. These are some of the common species of the Plasmodium which we usually come across. The important ones are Plasmodium falciparum, Plasmodium malaria, Plasmodium vivax, Plasmodium ovale, and Plasmodium novelsi. This is the life cycle of the Plasmodium, so that for that completion of the life cycle, they once need to attack the host. Here you can see various steps are given how they are doing the reproduction sexually and they are doing asexually. This is the diagram of Plasmodium falciparum under the microscope. This you will see or observe under the microscope whenever you are studying the slides of the Plasmodium. Here you have to prepare the thick smear and thin smear, right? Smear from the blood sample and you will find the Plasmodium. This is how you will observe it if it is present inside the blood. Next protozoa which we are going to study is the Paramecium. Paramecium is always characterized by a slippery shaped organism. We from the childhood days we know that if it is a slippered shape it would be Paramecium. So this is a very well structured diagram of the Paramecium with all the labeled parts. Contractile vacuoles are there, radiating canals are there, micronucleus, cytoplasm, food vacuoles, right? Buccal overture, everything is present in the cytoplasm. Paramecium is the genus again of unicellular ciliates, right? Paramecium are widespread in freshwater, brackish water, and marine waters. They are often very abundant in stagnant basins and ponds. The water which is not flowing, there you can easily observe this Paramecium from the sample of the water, right? They are doing the movement with the help of the cilia which are present in its structure. Various characteristics are again, they are widely distributed across the globe. They are mostly found in the stagnant water and their outer body is covered with the hair-like structures which are called the cilia. Now, how they do the locomotion with the help of the cilia, they keep on doing the locomotion, they rotate itself from the axis point. Then, what is the nutrition mode of the nutrition? Mode of nutrition is holozoic. See, in journals, you just have to mention the important characteristics, how what is the morphology. Here, I have given something extra so that you can easily understand about the organism, right? In actual practical you just have to observe the slide, draw the structure what you are observing inside the slide, and then you need to write the morphology and state the result that the which organism is present in the particular slide. The undigested residue is ingested through the temporary anal pore. We have seen in the figure that anal pore, so it is helpful in the digestion processes. This is the structure or figure showing the binary fission in the paramecium. So from this point, you can understand that the mode of reproduction is the binary fission, which is same like the amoeba. The common species of Paramecium includes Paramecium aurelia, Paramecium caudatum, Paramecium woodruffi, and Paramecium trichum. So these are the common examples of the Paramecium. This is how the structure you can see under the microscope. If you are observing the slide, you will see such type of structure. When you want to label, you can label it like this. This is one of the method how you can cultivate the Paramecium cultures. You need to take the grass, right? Normal grass which is available. Then you have to take the pond water. You have to place it in one bucket or a beaker after, for some time. Then rice grains, and then with the help of magnifying glass or with the help of microscope also you can. This is the way how you can cultivate Paramecium in the lab conditions, right? I hope you all are very much clear with the practical of microscopic observation of the various protozoa. Here we have discussed three major protozoa, that is amoeba, plasmodium and paramecium, right? There are various protozoans, but for your syllabus, this much protozoans are, it's fine if you study this three, amoeba, paramecium and plasmodium. Still, if you face any doubts, any difficulties, you all can contact and you can ask your queries. I hope you all are clear with the practicals. What you have to write and mention is you have to just draw the structure of the protozoa. You have to identify it, write its morphological characteristics, write few general characteristics and one or two examples of each of the protozoa. Thank you very much for watching this video and understanding. Thank you.